All right. Hello, everyone, and welcome. Thank you for joining us for today's session, the first in our new webinar series titled Environmental Monitoring Considerations for Gene-Drive Modified Mosquitoes. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Our mission is to support and advance best practices and informed decision-making for the development of genetic biocontrol technologies to improve public health. To do this, we work to identify and address key questions, provide technical advice, and strengthen capacity and information sharing. An important part of the testing pathway for new genetic vector tools will be post-release monitoring for safety and efficacy. This webinar series will highlight current vector and biodiversity monitoring efforts across Africa and introduce potential methodologies and tools that could be used to support environmental safety monitoring following release of gene-drive modified mosquitoes being developed for malaria control. Sessions will take place weekly and go through the first week of June. Before we get started, we have a few housekeeping reminders. Please email events@fnih.org if you have any technical issues. Your video and audio will be off during the webinar. The presentation will run somewhere between 45 and 60 minutes. Feel free to submit questions at any time in the chat. Questions will be addressed at the conclusion of the presentation. All webinar recordings will be posted on the Gene Convene Virtual Institute and the Gene Convene Virtual Institute YouTube channel. Today's webinar is titled Innovative Genetic Control and Environmental Concerns: What is being done to ensure the safe use of gene-drive mosquitoes? And we are honored to be joined by two outstanding speakers. Dr. Hamidou Maïga is a medical entomologist at the Institute of Research in Health Sciences, or IRSS, in Burkina Faso. His research centers on strategies for controlling vector-borne diseases, including work on mosquito swarming, mating behavior, and genetic tools. Dr. Maïga has led numerous vector control projects targeting vectors of malaria, dengue, yellow fever, and Zika. He has international experience from his time at FAO, IAEA, Insect Pest Control Laboratory, and currently collaborates with the Liverpool School of Tropical Medicine on a project to build molecular research capacity in Burkina Faso. He is also actively researching the potential ecological impacts of gene-drive modified mosquitoes. Inoussa is a PhD student in medical entomology, also at the Institute of Research in Health Sciences, and a student at the University of Nasibani. Inoussa holds a professional degree in biological engineering and a master's in medical and veterinary entomology. His work focuses on biosafety and environmental considerations of innovative vector control techniques, including gene drives. He specializes in field data collection, mosquito behavior studies, insectary management, and molecular identification. Inoussa has contributed to research on malaria, dengue, intestinal parasites, and GMO containment through programs at ICGEB and the Burkina Faso National Biosafety Agency. Before we get started, a quick reminder: following the presentations, I will be moderating a Q&A session, so please feel free to post your questions in the Zoom chat at any time. And additionally, today's session is being recorded and will be posted on the Gene Convene YouTube channel shortly after the event. With that, I'll turn it over to our speakers. Dr. Maïga, if you could go ahead and turn your camera on and get ready to share your slides. Thank you.
Can you see my slides?
Looking good.
All right. Thank you very much. Thank you, Yasin. Thanks to FNH for this opportunity in sharing our experience. I'm sitting today with my colleague Towe Inoussa, and we'll be presenting our experience, sharing our experience with the audience. So it's a great pleasure to share our experience on innovative genetic control and environmental concerns, what is being done to ensure the safe use of gene-drive modified mosquitoes. This experience from Burkina Faso. We'll be going through today a quick background on malaria and tools that are used to fight against it and the limitations. A brief literature review on what's going on vector control and the consequences. And then we'll focus of our experience from Burkina Faso on monitoring and ecology studies that may be used one day to monitor post-release gene drive. And at the end, we'll be sharing also a project that we are involved in and that can be used also to monitor gene drive. So as a background, let's say we may all know about malaria, transmission by infected female bite of Anopheles mosquitoes. We have about more than 400 Anopheles species worldwide, and 70 species can transmit the plasmodias. And there are 7 dominant vectors that are members of Anopheles group, Anopheles nili, Muschietii, and Finestus in Africa. And in Burkina, we have mainly Anopheles gambiae complex, Anopheles gambiae, Kuluzi, and arabiensis, but also secondary vectors that Anopheles finestus and nili. So the current methods to fight against malaria are mainly based on tools against the parasite, including the vaccine that the rollout is ongoing in few countries in Africa, but also conventional tools like vector control, including indoor residual spray and bed nets. However, the the spread of insecticide the spread of insecticide used to impregnate those bed nets are currently showing the limitation of these tools and forcing or showing the need of innovative tools for vector control. And for that, we have new molecules of insecticides and the release of some mosquitoes like Wolbachia that are mainly used for dengue, not yet for Anopheles to the best of my knowledge, and gene drive. So gene drive is a genetic system that biases inheritance. So a particular gene has a much higher chances of being passed on, often close to 100%, instead of the usual 50% on Mendelian route. So when gene drive is implemented, there are 2 approaches, mainly population replacement, whereby releasing a mosquito that may not be able to transmit the disease, such as malaria, or population suppression based on the reduction of fertility or biasing sex ratios that can crush the population, reduce the contact between host and the mosquito, and thereby reducing malaria. So, however, given that gene drive is known as being very powerful, we may see some irreversible genetic spread that may raise environmental and ethical concerns. So, given those risks of unintended consequences, how can we ensure ecological safety? So why should we act now? We have a tool. Why should we act now? We can see that there's a direct or indirect cost of malaria on humankind, mainly in Africa, where more than 90% of the global cases occur, and more than 95% of deaths also occur in Africa. So can we afford to ignore this technology? Or should we also ignore the risk it carries? Someone said, with great power comes great responsibility. What can we do? So here, I would like to show a brief literature review on malaria vectors, competitors, and predators that were released. And a few of those that were carried out in Africa have shown that vector control may have unintended ecological consequences. So for gene drive may not be new, gene drive may also have the same thing. But how can we safely monitor those? For example, there are like uh, some tools like bed nets that were distributed in Kenya and that have led to the decrease on Anopheles species, Anopheles gambi, but an increase on Anopheles arabiensis. So there's also residual spraying in East Africa that reduces Anopheles funestus, but led to its replacement by Anopheles rivularium. So that could alter or that altered malarial transmission. There are a lot of discussion on those studies. At least there's evidence here, or kind of evidence showing that those tools, any vector tool, may have a kind of impact. Um, those studies also showing that even if mosquitoes are not living alone in the breeding site. So how can we leverage on those studies in the breeding site to monitor what's going to happen if we release mosquitoes? There are strong association between predators and mosquitoes. They are overlapping with some subspecies, and some competition may also occur within the same mosquito species or with different mosquito species with significant and non-significant degree. So there's also this study that have by the team of Marshall showing that monitoring needs for gene drive mosquito project. What are the lessons learned from vector control? It says like assessing monitor requirement. It's important because you need to assist with the effective introduction of gene drive system in the field, but also detect unintended spread of gene-drive mosquito beyond the trial site. We know that we're talking about transboundary issues, but also resistance mechanisms that may happen or a non-functional effector gene that spread within trial or and intervention sites. So how can we monitor environmental safety of gene-drive modified mosquitoes? We would like to share here few trapping methods that may be used to assess the biodiversity. If gene drive wipe out a species, what will happen with the bycatches? So this mainly to show that by identifying the bycatches, we may be able to monitor the environmental impact of gene drive. So what makes a good trap? There are key characteristics mainly on specific behavior and ecological preferences. Let me take an example. For example, if we go, we know Anopheles arabiensis is mainly living outdoor. If we use a specific trap indoor, we have no chance or little chance of catching Anopheles arabiensis in some areas. So it's very important to know what's the best trap, what are you targeting, how you can catch those bycatches, and then you can monitor whether the gene drive have done its job or not. So it's few characteristics. An effective attractant is important, may work for some species, for Aedes species, but also Anopheles. So we have like CO2, the human odor alerts, visual cues, environmental mimicry, resting site, breeding site, and where to place those traps. It's also important, depending on the height, depending on the behavior of the mosquitoes. So what is the mechanism? To we sack the mosquitoes? Do we use like an aspirator? Should we use like a sticky surface? So a trap also may be easy to use and maintain. So a simple maintenance collection used mainly based on available tools, for example, in low in Africa, for example, instead of building some trap that may be very expensive. The durability and weather resistance, depending on the robust construction made from material that can withstand outdoor conditions, because most of the mosquitoes are trapped outdoor in the field in harsh condition. Weather protection, the safety and ethical consideration. The trap should not be toxic. and we need to protect the volunteers and people that are helping to catch the mosquitoes in the field. Should be cost effective so that we afford, and there should be specific targeting specificity. I just took an example on Anopheles arbiensis versus Anopheles culusae or gambi. So on the adult stage, we may use beach traps that are mainly used for monitoring population densities. But here, the aim, as I was saying, is to assess the impact of mosquito diversity. If gene drive have wiped out a species, we may, based on the catches, we may see what's going on, and whether gene drive have impacted also non-target species. And this is a study that have been carried out in Burkina and Mali, whereby they show that adding heat to the BG trap may increase the catches. So this also can help assessing the bycatches. Similarly, for the BG Pro and the CDC lights, I'm going quickly, but all those traps can be used to assess the mosquito diversity. And here, given the challenges for human landing catches, that ethical committee may see some issues. So people are now moving to alternative methods instead for human landing catches. So this can measure human biting rate by different species. And again, once gene drive have wiped up few species, we may identify here whether the biting rates or the species have changed. And we came across the dragging method using a drone that may go and sample in difficult areas, and how this can track gene drive mosquito after releases, but also facilitate frequent extensive monitoring, but also like adapted to capture specific group of insect and helping monitoring effect on non-target organisms. So can this be a potential tool for gene drive mosquitoes and ecological management? This can be. So I've been presenting this, the swarm survey is a study that we really master in Burkina since decades. It's very easy to do. And we can also, we believe that we can use this to monitor the environmental impact. How? By catches mosquitoes. If gene drive is able to wipe up a species or have impacted another species, we can identify easily the swarms. We can collect them. We can identify them and know whether, for example, if gene drive is targeting only Anopheles culusae, we know the differences between the swarms of Anopheles gambi and Anopheles culusae. We know at which height they may be swarming. Then we can collect and give a feedback on what's going on. For the larva stage, there are studies identifying different species of predators, but also here, the objective is to collect non-target organisms living in those larval habitats. And this could be done in different period. It can be done as baseline data before the releases, during the releases, and post releases to monitor what could be whether we have like the gene drive modified mosquitoes that mated with white mosquitoes, whether the outspring like larvae or eggs are collected in the breeding sites. In addition, we know that Anopheles mosquito breeding sites, they are breeding in very small, depending on the species. They may also breed in rice field paddies, but they may breed in very small breeding site that may not be accessed easily. So how the use of unmanned aerial vehicles like drones and AI can be used to identify those and evaluate the changes in the population when gene drives releases, and identify habitat changes that could affect non-target organisms. So this also can be used for biodiversity monitoring and detect any changes in populations. So now I would like to share the experience in Burkina in monitoring and ecology. So we were involved in the project funded by the Foundation Bill and Melinda Gates Foundation to PAMCA, whereby we identify challenges that national malaria control programs are facing for entomological surveillance. And so we identify the lack of infrastructure, the skills of the personnel. And for that, data analysis skills, those were missing. And we thought like vector control has really showed its importance in the fight against malaria. So how can we support those people? And how can we use them to help monitoring across the country? So, and this we show that this was very important if we really want to target a malaria elimination goal in Burkina. So this project will run in collaboration with the national program, a national malaria program, control program named SP-PALU, which is elimination now, Secrétaire permanent pour l'élimination du paludisme. And they were very helpful in recruiting the personnel across the country for the training. So how we did, the project aim was to monitor the abundance of target species and secondary species across the country. So how the trained people across the country can assess, can collect the mosquitoes samples and ship them in the laboratory in Bobo-Dioulasso so that we can do subsequent analysis. And this, how could we use those to assess the epidemiological impact? The second objective was also to identify possible opportunistic competitors in the breeding site, because what I didn't say, that they were collecting adult and larvae, and in addition to the non-target organisms. So how this, knowing those can protect the ecosystem, avoiding any consequences when gene drive is releases, and also identify like whether they are like the predators, the competitors, or potential pollinators. So after the representative of National Malaria Program were trained in our lab, they go back and involve the communities and build those kind of local anti-mosquito brigades for monitoring purpose. And then in each period, whenever we want to monitor, to collect mosquitoes or immature stages, we contact them, we provide small equipment, and they proceed with the collection. So the methods on the training of those NMCP representative, we use adult catches using pre-treated spray catch, PSC, aspiration, the use of traps. So these are like the package of the training that we've done. We use with them. They spend like 3 to 5 days, like theoretical and also practical in the field. So the results, when they collect the mosquitoes in the field and they ship them, we are able to map the species composition across the entire country. And you could see that Anopheles gambiae on the left map. Anopheles gambiae is the main vector here. And this also allows us to identify one of the secondary vectors, which is Anopheles fenestus, which is mainly in the Transvaal region and the southwest of the country. So this data we found very important in case we'd like to release gene drive, how those data across the country may allow us to know whether suppressing Anopheles gambiae could lead to the invasion of secondary vector, which is Anopheles finestus. We also said before that the studies that we showed can also demonstrate that ecological niches left vacant may quickly be colonized by other species. So in our case, if we wipe out Anopheles gambiae, Anopheles culusi, Anopheles finestus may take over. So monitoring based on what we have should be multidimensional, combining genomics, population ecology, and epidemiology. For this, what I didn't say, when we collect the mosquitoes and they are morphologically identified, we also go for molecular and also sequencing to make sure that we have all the information that we have in the field before any gene drive or any, and if any other technology is used, we may see the changes and the impact on mosquito species. The monitoring should also be anticipatory, using distribution maps and spatial and temporal dynamic to predict area of risk of replacement. We could see the distribution of Anopheles finestus mainly is located in the country, central region and southwest. So any change in the next coming years or after release or any technology, including gene drive, we may have an idea of what's going on. So we believe that this can be used. So the monitoring should be also inclusive, involving local communities in data collection. That was very useful, very good experience. And forming local anti-brigade was very important and very, very nice to have. So this being said, I will stop here and give the floor to my colleague Inoussa, who will be going through the research on immature stages that we have done and that may be used for gene drive monitoring. Thank you.
Can you hear me? Good afternoon, everyone. So thank you to Dr. Maïga for giving me the floor. Following the insightful presentation on adult stage of monitoring of gene drive modified mosquitoes, I'm delighted to delve into the critical yet often understudied aspect of immature mosquito monitoring in Burkina Faso. As we know, the ecological interaction at the larval stage are fundamental to understanding the potential environmental impact of gene drive modified mosquito, particularly in complex natural habitats where Anopheles gambiae thrive alongside non-target organisms. Today, I will share some key activities from Burkina Faso, focusing on two main aspects. The first one will be on the non-target organism diversity in Anopheles gambiae larval habitat, who is essential for assessing ecological risk. And after that, I will present the protocol on the larval competition dynamic between Anopheles species and co-occurring species could influence gene drive spread and mosquito population structure. So the main objective of the monitoring of the immature stage is to monitor and assess the diversity and the abundance of the non-target aquatic macroinvertebrate coexisting in Anopheles gambiae SL larval habitats and to study their interaction, to analyze the spatio-temporal dynamic of communities of non-target organisms, to understand the seasonal variation, And finally, to explore competition between species through niche overlap, co-occurrence pattern, and the influence of physicochemical parameter within Anopheles gambier and other mosquito and macroinvertebrate in natural larval habitats. So before we can assess the ecological risk of gene-drive modified mosquitoes, we must first understand the natural biodiversity in the larval habitat. In Burkina Faso, for example, our team conducts extensive sampling of macroinvertebrate communities in Anopheles breeding site to identify potential non-target organisms that could be affect. So let's explore what we are doing in Burkina Faso. So here we have a study design. So our sampling strategy encompasses 70 districts across the country to ensure optimal ecological representativeness. So site selection was based on 3 criteria: larval habitat density, spatial distribution, and diversity of available habitat types during the study period. So for each field campaign, we systematically sampled 10 representative larval habitat to sample at each survey. And particular attention was given to precise site documentation using GPS recorded coordinates in decimal degrees. And I can say this standardized approach enabled us to obtain spatially and temporally comparable data while accounting for natural variability in Anopheles colonized aquatic ecosystem. So this methodology is rigor and can allow us to establish here a robust foundation who will be maybe necessary for the subsequent analysis on the non-target organism and also for the larval competition dynamic. Here we have our sampling site. As you can see, we did the sampling across all the country. So for the field sampling, equipment and protocol, I can say that all the sampling sites were documented using GPS device, recording exact latitude and longitude coordinates for reproducibility. And for each breeding site, depending on the size of the breeding site, we did like a specific number of deep. So for the small habitats, as a house spring and a small puddle, for example, which is inferior to 1 meter, we took 2 scopes. And for the medium habitats, which is between 1 and 5 meter, we use 4 scopes. And for the large habitat, which is superior than 5 meters, we use 6 scopes. So we use also the sieve mesh to isolate and separate the mosquito just after collection to the non-target organism, and we preserve them in the ethanol, 90%. So all the sample collected were labeled, and I can say that the label include the site ID and the habitat detail and the date as well. So here I can say that systematically each habitat that were made during our study was systematically inspected, and we map the microhabitat zone as age, vegetation, shaded area, And for each breeding site, we conduct like 3-minute visual scan per zone before physical sampling. And we recorded also the absence data, which is really important, I can say critical for distribution model in template. So for all the specimen or non-target organism, macroinvertebrate that we collect, we use like a dichotomous case from Robert and all 2022 and Russell and all 1945. And I can say that for the good practices, reliable identification is record at least 2 independent cases. Always document ambiguous character for future review. Integrate the morphomolecular approach when possible. So here, I just wanted to show you several non-target organisms that we meet during our study. We have like Edisticidae, Corycidae, Libellulidae, and Hydrophilidae. So this photo illustrates that the different NTOs of stress fraud are our study in our field in Burkina. So here I'm going to share with you the breeding site that we meet. So we meet like 7 category of breeding site. The first one is the artificial breeding site, which are the bucket, the tard, and the clay pot. So we have also the tire track, the puddles, the rice paddy field, the pond, the stream, and the river pocket. So just at my right, I think that you can, it's also at your right. So we have like a Sankey plot who shown, who provide a valuable insight into the relationship between the breeding sites or the larval habitat and the different species that we found and the interactive, the relative abundance and distribution of each of them. So for instance, the white of each flow represent the strength of the association, meaning larger flow suggests a higher abundance of a species in a specific habitat. So Anopheles mosquito, our study just shown that Anopheles mosquito colonize the puddle, the pond, the tire track, the river pocket, the stream, and the rice paddy field, rather than the artificial habitat. So additionally, it's important to note the presence of the macroinvertebrates such as Betidae, Corycidae, Nepidae, Hydrophilidae in the same habitat as Anopheles mosquito, even if they were deficient in abundance, I can say. While documenting NTOs or non-target organisms is crucial, we must also consider how gene-drive modified larvae or mosquito might interact with wild mosquito population and other species. So competition for resources at the larval stage can significantly influence mosquito population dynamic and by extension, the spread of gene drive. So our study, we are going to show you what is doing in Burkina to assess the larval competition. So we conduct this study in 3 sites, 3 villages, which are Bama, Sumuso, and Pala. And for the designs, 3 distant periods were targets, so the beginning, the middle, and the end of the rainy season. And for each breeding site that we made, we can say that we recorded the GPS coordinates and also the physical-chemical parameter as the pH, temperature, the percentage of oxygen, the turbidity, and the conductivity. and we assess 15 breeding sites per site and per period. So to summarize, I can say that if gene drive can reduce the target population, it could lead to a reduction in the competition for breeding sites and an increase in the density of other competing species, such as secondary vector, can be Anopheles fanestus and Anopheles nili in Burkina, and other genera who are Culex Aedes mosquito. So restricting that can also restricting the aquatic community, including macroinvertebrates. And the most important things here is that all data on the interspecies competition can be integrated into ecological and population dynamic model. And this may help us to stimulate gene drive introduction scenarios and to better prepare the control release phase and post-release monitoring to anticipate ecosystem impacts, to adapt also to monitoring, and to inform for decision making. So I'm going now to present to you and integrate environmental monitoring tools that we may be use in Burkina also for the gene drive post-release monitoring. And I can say for this project, we in collaboration with a teams in CIRAD, especially with Dr. Jérémy Bouillet and also Dr. David Wilkerson. So I can say that there is a lot of job. We demonstrate that eDNA is a versatile and sensitive tools for detecting and quantifying and molecularly characterizing emerging vectors, as well as invasive species or immature stages. But I just want to highlight this study for Antoinette Paggio, who is also part of this webinar series. She is going to go deeply on these tools, the eDNA, how the eDNA can be used to detect the gene drive construct. And I think that in this study here, she shows that we can use the eDNA to detect the gene drive in musculus. So how can eDNA improve surveillance of gene drive impact ecosystem? Here we can say that eDNA can be used for biodiversity monitoring, gene drive tracking, non-invasive detection, and this method is a scalable method. So here we have like a kind of application of eDNA in gene drive monitoring. And for example, for the tracking of the gene drive spread, we can see that eDNA can allow us to determine or to detect the presence of the absence of the construct, and then to allow us to monitor the population. We have also another application on the establishment of the ecological baseline data. So we can use eDNA to establish the pre-release data and also the post-release data so that we can maybe measure the potential impact of the gene drive release. So we have also the biodiversity impact assessment. We think that eDNA can be used for predator and competitor monitoring as well. And what else? Maybe Dr. Antoinette will come back later with the eDNA deeply. So here I'm going to present the balancing potential and limitation of the eDNA. So for the advantages, we can say that eDNA can provide a high sensitive detection, is non-destructive and cost effective. For the challenges, we can notice that the possibility of the false results, it's depending on the database, and it's true that it's not here, but I can say that with the eDNA, we have also a problem of a quantification. So we cannot know exactly which quantity of maybe species is in this breeding site or another one. So here for the case study and future direction, so we have seen in the previous slide that eDNA can be used for gene drive detection. So I think that as I know, there is for the moment no study that's shown that eDNA can detect the construct in the mosquito population or in another genetic control tools that may be the SIT. So we think that we may be to standardize the protocol. I just want to say that here, like we may need to develop and refine eDNA protocol for consistent monitoring of the mosquito, and to see how is it possible to combine eDNA with remote sensing and citizen science for comprehensive data. So I can see taken together from NTOs diversity to label competition and eDNA monitoring paid a comprehensive picture of the ecological consideration for gene drive deployment. As we move forward, integrating these insights will be a kind of key to ensure that gene drive technology are both ecologically sustainable and socially responsible. So I cannot maybe finish my presentation without acknowledge my mentor, who is you can see on the last slide Professor Diabate, and also Dr. Professor Jeremy Bouillet. So thank you for your attention, and I think that we are available for the question session. Thank you.
Thank you very much, Dr. Maïga and Inoussa. That was some very interesting points you guys raised. So I see we have, it looks like now we just got a second question in the chat. Reminder to the audience, if you have any questions, please go ahead and post them in the chat. I believe if you have a burning question you need to ask yourself, go ahead and raise your hand for that. But let's go ahead and get started with the first question, which was, I had a very similar question, so I'm kind of going to hybridize them and combine them. And that was, I would like to know, faced with the challenges of capacity building following your experience in Burkina Faso, how the use of tools such as drones and AI can be efficient, or I think probably increase efficiency. And my question on top of that was, what role do you imagine AI playing in the future of environmental monitoring, and how could it play a role in malaria control capacity building? That was a mouthful, so I'll give you a second.
Thank you very much, Justin. Thank you, Inoussa, for the great presentation. Yes, thank you for the question. I will try to answer, and Inoussa may add on if necessary. So the use of new tools, and including drone and AI, how they can. I have no knowledge on not great knowledge on AI, on the use of drones a bit, in environmental monitoring of mosquitoes, or also releasing mosquitoes using drones for better dispersal, and in areas where, for example, for Aedes mosquitoes, in highly dense population, it's very hard to release by car. So releasing with drone may be a solution. But here we mentioned the use of drone to see the type of breeding site that we have for Anopheles mosquitoes. They are scarce and very well dispersed. So a drone may be mapping, giving a distribution of those water bodies that later on we can go with the precise GPS coordinate. We can go and then monitor, mainly when there is a very rare number of breeding sites in the area where we want to monitor. To add on the AI, so AI is a very good tool that today we are also trying to catch up, because it's very fast. And I believe that with those tools and the massive data that they can gather may lead maybe one day to AI custom tools for field technicians. This may ease the collection in the field and in the real time, because even without AI today, we may use uh, smartphones. We can gather some data. If we found some species in the field, for example, we can take a photo, and then it's stored in how to say. I think we are using Kobo tool. My colleagues, geographer Aziz Milogo, would be better to answer on that one. But these tools can be really used to make it more efficient and gathering more data. So this is what I can say on that.
Very good. Nusa, did you have anything to add, or should I move on to the next question?
To ajouter quelque chose. He said he's good.
He's good. All right, let's move on to the second one.
Okay.
Given how variable eDNA data can be, how will you attribute any change in non-target organisms to a gene drive? How large would the effect size of the gene drive induced perturbation have to be for you to see it? And have you carried out any sort of simulation or power analysis to detect it?
Thank you for this question. As Inoussa has presented, this is a new tool that we did not assess yet in Burkina Faso. But we are keen in learning, and Inoussa is supposed to travel to Jeremy Bouillet's lab for the training in collaboration with David, so that we can use it one day. So I cannot say anything on that yet. So, and I believe that there's a lady that will be presenting on the eDNA. That question she may be the best fit to reply to that. Thank you.
Very good. Yes, we have, towards the end of the webinar series, we have two presentations on eDNA. So we'll hopefully get some answers at that point. Let's see, the next question. How do you manage to extract eDNA? Another eDNA question. And is it the same protocol as for extracting DNA?
So I think it's a similar question. Like, we have never tested yet here in Burkina. So there are a few papers, and I think on the presentation we shared some papers that have done it. The first paper that we saw, I think they assessed how Egypti and Stephensii may breed, and how using this non-invasive method to detect species, invasive species, it was great. But we have never assessed it yet. So
right, so future work.
Yeah, thank you.
All right, the next question is, how long do you think you would need to conduct baseline non-target organism monitoring to detect a meaningful change after release of a gene drive mosquito?
Justin, can you repeat that, please?
Yes. Let's see. How long do you think that you would need to conduct baseline non-target organism monitoring to detect a meaningful change after release of a gene drive mosquito? So for how long of a period would you need to do ecological monitoring to detect a change, you think, in your best estimate?
That's a great question. I would say maybe I will extrapolate with what I've done in the field for mosquitoes. We assess, for example, the mosquito dynamics over 2 years to make sure that we see the fluctuation in mosquito densities depending on the season. Because here in Burkina, we have like 2 seasons, like dry season and wet season. So running over 2 years, we are sure that we have enough data to show what's going on in the field. And so I believe any assessment of other tools may follow that protocol until the protocol shows that it's not very meaningful or it's not working for that specific thing. Thank you.
Yes. All right. The next question is a niche replacement question. You have shown that in the event that gene drive is deployed to suppress Anopheles gambiae, the niche will be filled by other mosquitoes, including Anopheles fenestis. What would be your recommendation? So I guess provide a comment on your thoughts on niche replacement or having one mosquito species replacing the target of a gene drive.
Inoussa would like to take that. On the niche replacement.
All right. Can you hear me?
Sure can.
I think that this is a question of, I can see Mr. Sawadogo. Okay. So as I can say, we are going to assess, so first, the niche overlapping between the mosquito species. And in case that maybe there is a kind of competition between Anopheles funestus and Anopheles gambiae, that's we may maybe, how can I say, let us know that in case Anopheles gambiae will be maybe suppressed, Anopheles funestus will take the lead. So that can provide us like a clear information that we may pay attention to the secondary vector, which is Anopheles funestus. And I think that most of the study currently are looking how is it possible to target also the secondary vector, because the male is a male. I think that Anopheles funestus in Burkina Faso is a male secondary vector. So we are trying also to to study, to run some kind of activity on this specific vector, so that if we just we have like a kind of information about the competition, we can also target Anopheles funestus to avoid this cascading effect and to maybe suppress also Anopheles funestus. So that will depend on the strength of the competition between Anopheles funestus and Anopheles gambiae as well.
Thank you very much, Inoussa. I see we have, this looks more like a comment, and it says, AI deep learning can identify breeding sites on high resolution drone imagery. So that could be a potentially useful tool with respect to AI and drones. I have a few more questions that I might ask, and then we can close out if we don't. Oh, we got another question in the chat. So let's see. In your opinion, with climate change, what will be the major challenges for vector control in the next 5, 10, or even 20 years, broadly speaking, by 2050?
So with climate change, what will be the major challenges for vector control in next 5? So I will take that one, and Inoussa may add if possible. What we know is like climate change may drastically change the condition in different areas, places where the condition were not conducive for mosquito breeding, maybe or not. So we believe that if we are monitoring, in our case, areas where we know the distribution across the country, and then based on that, we would like to design a control tool in the next coming year, 5 years, for example. If there is a drastic change, meaning that any control tool that we'll be implementing may not work. And the reason is that maybe the temperature or um, the rain, the quantity of rain has allowed some mosquito or other species to grow and transmit. So I think temperature, changing the species distribution across a country, across a continent, may be something that we need to look at. Inoussa?
All right, I think that I can add something. So we know that climate change can really impact, can really be a challenge for vector control. That's why I think that Dr. Maïga was talking about the projects, the Welcome Trust and PAMCA, and also Bill and Melinda Gates about the capacity building. And I can highlight the thing that the data analysis is really important. And in Burkina, we have a lot of data, and we think that we associate also the is a geograph to collect like a climate data so that we can use like a model to see right now for maybe when we go back in the previous 10 years. So what was in Burkina, and right now, what is the kind of changement? So we can assess with a model to see and to predict. How can I to predict? Yes, the potential. I can say changement in the vector dynamic for maybe the next coming years also. But it's true that that can be a really big challenge for vector control tools.
Yeah, very good point. I imagine, yeah, change, especially changes in rainfall, could. I loved your graph where you showed all the habitats and what you collected there. Changes in rainfall could really impact where you might see mosquitoes. So that was a really great question. Um, let me ask this question since I don't see another one in the chat. Um, one of the questions we see a lot has to do with data portability. So if you collect data in Burkina Faso, could you use that data in Kenya or Uganda? How might the baseline data from Burkina Faso inform gene drive risk assessments in other ecological or geographical contexts?
So good question. I can try. Um, yes, this is great because what we may not, depending on where we are, we may not have the same species. And so the tools that we may be using or gene drive targeting, for example, Kuluzi may not work for, I don't know, Funestus. And so I don't know what this can impact Kenya, for example, or another country where Funestus is, why is the gene drive is developed on Anopheles gambi. But what we know with the gene drive is that there's a challenge of transboundary issues. And so if we want to release, for example, in Burkina or any other country, it would be important to know and train people across like the neighboring countries at least to collect some data, and then we see whether there are changes or how far the mosquitoes, how gene drive mosquitoes are traveling. So I may say that for neighboring countries, it can be very informative, and more especially if we are sharing the same species of mosquitoes. But again, it can really change, and only data from those countries can also try to. But what I can add, because we are talking a lot about modeling, with modeling, we have data from Burkina. We can take scenarios. If I'm not wrong, I'm not an expert in modelization, but how data from Burkina for several years with known species, we know how it breeds, how this can inform a program in Kenya or in any other country. Modeling.
Modeling.
Thank you.
Of course, thank you. We got another question in the chat, another one on eDNA. Can you explain a little bit how eDNA works and how it can be efficient for monitoring of gene-drive target site and natural populations? How does it differ from amplicon sequencing, which can be easily used to assess genetic variation at a given genomic region?
I would love answering that if I was a molecular biologist and if I had hands-on on that. But Inoussa, do you want to say something on that? So I think the next coming webinar sessions on eDNA, there will be a lot of interest, which is good. We'll be also attending to learn more on that.
Yeah, I think eDNA is an emerging field, so I think there's a lot of things to be sorted out, and stay tuned. And I think we definitely have some eDNA specialists that are going to come in and talk a little bit more about how it works. So with that, I don't see any other questions. Maybe I'll ask one more myself, just because you kind of hinted at it in one of your previous answers. How scalable is your current approach for other countries or regions where gene drive mosquitoes might be considered for release? What changes would you make to your process, protocols and such, to increase scalability? So how transferable what you're doing now is it to other countries, and what might you change to increase scalability?
So thank you for this question. I think the method is like, I think most of the countries in West Africa are using the same scenario. I mean, the Minister of Health, in collaboration with Minister of Research, they have like what they call sentinel sites across the country, where they have like contacts that can monitor and collect some samples that they can share. So the thing is, the current method is using people need to be trained, and then they will train others. But nowadays, I know that in Tanzania they're using kind of like how they call it. They're using like the smartphones. People can use it easily and then share some photos, those kind of things, and also collecting, I believe, so that they can assess whether what people are sharing it's exactly there. So if at some point we may not need to collect the mosquitoes and do some like sequencing or molecular tools, and based on the photos that people across the country or across the city may provide is enough, then this will make things easier, because then we have more people involved and they can easily contribute. So this is something I think great that we may add on what we are currently doing.
Yeah, that's fantastic. I think that kind of crowdsourcing people to help out with it is a fantastic way to increase scalability and get stuff done. So I don't see any other questions in the chat, so we can go ahead and close out here. Everyone give Dr. Maïga and Inoussa a round of applause. I know we can't hear them, but I know everyone's clapping. I can hear it off in the distance. But thank you guys so much for your engaging presentation, and thank every one of the attendees for joining us today and contributing to the discussion. We hope that you'll join us next week, same time, same place, for our second session in the series with Dr. Fred Boyager Antwi, who will be presenting on an ecological observatory study of Anopheles gambiae. As a final note, we invite you to visit the refreshed Gene Convene Virtual Institute, where you will find new resources, tools, and publications. And while you're there, consider subscribing to our newsletter to stay informed about upcoming events and activities. Thanks again to everyone that attended. Thanks again to our speakers, and we hope to see you next week.
Thank you. Thanks for having us.
Thank you.
Thank you.